This webinar is being recorded and it will be available for viewing later on the EAB University page of the www.emeraldashboard.info website. Thank you today for attending. And Janna, you are now unmuted and begin your presentation.
Hi, it's good to be here. My name is Janna Beckerman. For those of you who don't know me, I'm the Extension Ornamentals pathologist amongst several other crops here in Indiana at Purdue University. I was told to give a really brief bio, so as brief as I can do it. I was born and raised in Cleveland, Ohio. I went to school at the College of Environmental Science and Forestry and finished up a PhD at Texas A&M. From there, I went to the University of Minnesota before I ended up here. So I've really been as far south as Texas and as far north as Minnesota. So I feel like I've covered a pretty good geography, and then as far east. So I'll pretty much be talking about sudden oak death and its ramifications here for those of us who live east of the Mississippi, which is a lot different than most of the discussions that I think have happened to date. So with that, I'm going to just get started and start with, the name sudden oak death and what it is. Well, what it is a disease that was named because it was first identified as being very deadly to oak and to tan oak. Since then, we've learned it's actually pathogenic to a number of different host plants. It's in the several hundred and counting. And unfortunately, sometimes the symptoms show up as a minor leaf or shoot blight, and in other instances, it can actually kill the entire plant. So it's caused by Phytophthora ramorum, which is a pseudofungus, and I will be talking about all of these things, but I'm trying to put this in a context of the disease triangle. So you'll have to picture me in front of you making the little triangle symbol, which is, just something that all plant pathologists have to do in every talk. And when we talk about any sort of plant disease, if you get nothing away from this talk than this, please take home the fact that you need this interaction between a virulent pathogen, which we certainly have with Phytophthora ramorum, a conducive environment, which we're going to talk a little bit about, and then a susceptible host. And what's unique in this disease is that there are so many susceptible hosts, and susceptibility spans from leaf spots all the way to the death of the host. You need all of these things, though, to come together in order for the disease to occur. So we're going to start with the host, and unfortunately it's too numerous to list. And because this disease came out in the West Coast and first appeared there and was first identified, most of our information actually comes from the West Coast. And we've been in a bit of a catch-up mode and just trying to make sure it doesn't get established east of the Mississippi. So Phytophthora ramorum on the West Coast has been associated with this list here primarily. They talk about a filthy 5, but as you can see here, rhododendron makes up almost 3/4 of all the reports. And so this is really, the filthy first, and the one host plant we really want to keep in mind in the nursery industry and in the landscape, because this is where kind of all the Phytophtheras go to party, and they love their rhododendron. So the problem with sudden oak death, as I've mentioned, is the wide host range, and unfortunately the name sudden oak death. The other name, ramorum canker and blight, hasn't really picked up the way it is, but here you can see an image from California that it is obviously affecting oaks, but it's affecting a lot of other trees as well. And this is why we should all be concerned, is we've seen the incredible damage that it's done on the West Coast, and we are greatly concerned about all of the hosts we haven't yet identified, potential hosts here on the East Coast. So on the East Coast, some of the hosts that we need to be concerned about, these are just a few that we picked up. Some of the big ones obviously are similar in both coasts. Magnolia showed up on the list in addition to rhododendron and azalea, prunus species, which are so popular in the landscape, and Pyrus and Kalmia, which are more, I think of as the Carolinas and the South up the eastern seaboard. All of these are really good hosts. my sense, and I really don't wish to see this experiment go into play, is that if something is a host to Phytophthora, it's probably going to be a pretty good host to this Phytophthora. It might only be a foliar blight, it might cause the death, but it is something that you really want to keep in mind when you're dealing with this out in the landscape or even in your nursery. And I say this because of this image here, and this was a photo, as you can see, taken in 2015, and this is larch that was infected with Phytophthora ramorum. It is now widespread throughout the United Kingdom, and before anyone says, Janna, larch loses their needles regularly, you can see the early stages of the infection here and here. And the other thing that's just so dramatic is how quickly this has spread from the first recognized identification in 2010 to where we have it now in 2018. Now, one thing about the United Kingdom that's very different than those of us who live in the continental Midwest or eastern half of the United States is the level of rainfall. But that very much is something that we want to keep in mind, along with the incredibly invasive nature of this pathogen. So going about this invasive nature, we can see here it was first introduced, they believe, in California. There was subsequent introductions into British Columbia, and it went up and down the West Coast. We have had several other introductions in the course that have managed, to our knowledge, been mostly contained. We do occasionally seem to have reports out of, I believe it is South Carolina and Georgia, where they have actually baited out Phytophthora ramorum, so it seems to be there, but they're not quite sure what's going on. This kind of came to a head again this year because in 2019, there was an identification and confirmation in those states with stars you can see here of Phytophthora ramorum, and these were released in a big box store from rhododendron. So they were actually in play for a while. So not only were the rhododendron infected, but it is absolutely possible that other plants were infected and ended up being planted out in the nursery. Our concern is when you superimpose this the map of the releases on the map of what our risks for sudden oak death are. For those of us in Indiana, we have 17 species of oak. You can see in the southern part of our state, the wetter part of our state, it is considered a high risk. Obviously, those in the Great Smokies and in Appalachia and in the wetter mountainous regions that are often cooler are at the highest risk right now. So these are things to keep in mind. And just to reiterate, I mean, this is a problem that's worldwide. This is one instance, as you all know. FedEx is bringing absolutely everything overnight, including a lot of invasive pests and pathogens. And I'm not into the blame game here. It's just something that we really need to recognize and we need to work with, whether it's happening within the United States or if it's happening in international plant trade. So going back with this pathogen, why should we be concerned? I hope I made that very clear. The fact that it has so many different hosts, it even has so many more potential hosts, and the fact that it can easily spread and sporulate off of many of these minor hosts and end up causing the death of what we would refer to as perhaps climax species hosts, such as oaks, beech, and maple in the forest. Phytophthora, for those not familiar with Phytophthora, it literally means plant destruction, and I think I've shown you the photos there. For those of you of Irish descent, this pathogen, this genus of pathogens had a lot to do with your family's emigration from Ireland to the United States or to Australia. It ended up directly causing the death of 1 million and the emigration of about 8 million other people. So this is a pathogen genus that has a history of tremendous destruction. This can also be witnessed not just with sudden oak death, but Phytophthora cinnamomi. In Australia, the eucalyptus forest is referred to as the Jarrah. And to give you an idea about some of the potentials that are of concern, in the Jarrah, up to 45% of the trees and up to 50 to 75% of the herbaceous perennials were actually killed. This has resulted in the extirpation of many different species and many more that are severely endangered because of a loss of wildlife mast, food for wildlife, cover, and the situation there. And here you can see what it looks like now. Phytophthora cinnamomi is a very important pathogen of avocados. If you were wondering why your avocados are getting so expensive, it's actually because of two invasive diseases, Phytophthora cinnamomi and laurel wilt. And here you can just see an overhead infrared showing, again, the ability of this pathogen to spread. And here's one final picture. This is a Phytophthora that has yet to be identified. This is in a nutmeg plantation in Grenada. And these are pathogens that not just affect our landscape, they affect our food, they affect our fiber, they affect every aspect of human existence. Here in the eastern part of the United States, we have a lot of issues with Phytophthora citricola and pleuravora, depending on how it's identified or misidentified in my case. And you can just see the progression that results. So these are all things to keep in mind when we start to deal with Phytophthora. So focusing in on the pathogen and what it is. So we've talked about the host and how it spreads. The pathogen, it's really important to stress that this is not a true fungus. We can call them water molds, we can call them fungal-like pathogens. I can impress you with wonderful words like stromanopola, which I defy anyone to say 5 times fast. The key point about this is the cell wall is more like a plant. It's made up of cellulose and less like a fungus. And it also shoots out these little swimming zoospores. So very different than other fungal pathogens. As far as what it's related to, its cell wall is more plant-like, but it's actually more like protozoa. And by that, these would be like your Daphnia and other protists that you will find in water. I know this is like a lot to wrap your head around, and I don't want to get too into this. I just want to make it really clear that this is not a standard fungus, and that many of the fungicides that we use for, say, fusariums or colototricums or rusts or, apple scab aren't going to work to control Phytophthora. As a plant pathologist, we have to discuss the life cycle, or as best we understand it, with Phytophthora ramorum. We'll start with the oospore, which is the product of sexual reproduction, which we think germinates. It then goes on to produce possibly a sporangium and motile zoospores, which will actually swim. These zoospores can be baited out of rivers and streams, even on the East Coast. So we do know this actually exists. We just don't actually know where this is overwintering and if it even is actually having sex because of the limited number of introductions. You need to have, obviously, for lack of a better description, a boy and a girl, and the number of introductions to date haven't been consistent as far as if we're spreading boys and girls, for lack of a better. When we think about Phytophthora and what they are oftentimes root rots or root munchers. And what it does is just reduces the root volume. And this is Phytophthora cryptogea. And you can see it just makes a stunting. So this is, something to keep in mind. Ramorum is not so much affiliated with this, but other Phytophthora are. Ramorum is affiliated with these crown rots, and you get these bleeding cankers like you see here. Oftentimes you might actually see, because people look at the foliage before anything else, you might see what appears as scorch, and this is simply because the damage underneath the cambium on the stem of the tree is preventing water from reaching the upper canopy, showing symptoms of scorch. If you excavate this away, you'd actually see these discolorations underneath it. And again, this is all Phytophthora, and I really want to stress that you want to make sure you don't just assume that something is a citricola and not a ramorum, that we treat all Phytophthora the same way. In the nursery, you get crown rots that look like this, and again, you'll get some of this scamosis and weeping, particularly on your prunus species. Here's another Phytophthora on causing a foliar blight. In this instance, this foliar blight is caused by Phytophthora citricola, but it certainly would have been one of those things to consider, Phytophthora ramorum, if there had been a report of release of ramorum at that time. And here's something that looks incredibly inconsequential, and this is on Viburnum davidii, I believe. And it's simply just a leaf spot. And this is where Phytophthora ramorum is so deceptive, because this actually is Phytophthora ramorum. This was a photo I took out in British Columbia. And so you don't want to assume just because it doesn't look that bad that it's not Phytophthora ramorum. There is a lot of symptom variability here with this pathogen. So not only do they cause root rots, crown rots, foliar blights, but they can cause all of the above, and that's very common in the greenhouse industry. You will see these types of foliar blights here. But one of the things to keep in mind is the fact that you can see here that in the case of this foliar blight, it ended up jumping the shark and also infecting these plants behind it. So this is really, again, the awesome powers of Phytophthora, and why they're, for lack of a better word, kind of funky. They're not textbook in many ways, in my opinion, that you have this really broad host range with such a diversity of symptoms. All right, so we've talked about the host and the pathogen. We really have to go and discuss the environment briefly. Phytophthora like it wet. And so this, to my mind, I've called it this before, I will call it this again. This is like a brothel for Phytophthora. They are completely partying down here, and they love this situation. You have a multiple of hosts. You have not only wetness for any sort of foliar blight, but you also have them in standing water. You can see the algae here, that this has been a long-term problem. There's some leftover material. There's, not dying, it's more senescing material, but this would be a really happy place if you were a Phytophthora. And so we want to keep in mind that, wetness is hugely important, but at least for ramorum, this cool wet soil is essential. Most of the Phytophthora we deal with are going to be more warm season Phytophthora. So that's something to keep in mind if you're starting to see, if you're in an area that is consistently cool and wet, like you do see in the mountains, or my concern is for those of us who lived in the Midwest this last summer, it was tremendously wet. The other thing to keep in mind is the fact that with Phytophthora, the symptoms oftentimes show up long after infection. So you can have infection, and as long as the tree or shrub is somewhat moist, wet, you're not going to see symptoms of wilting or decline. And it's only when it gets hot and dry that the plant will completely collapse. The infection didn't suddenly occur. It's that the plant was suddenly no longer able to provide itself with the moisture that it needs to survive. So what's going on with the environment? Well, obviously flooding is a huge issue. This is something you're going to find a lot of Phytophthora by streams and ponds, and for those of us who live on heavy clay soils. I'm using some sort of extreme examples here, but obviously any of these situations are going to be conducive for Phytophthora. So then how do we diagnose it? And this is one of the things, when I started as a grad student far too long ago, we were just happy to identify something as a Phytophthora, and we would leave it at that. This was before Phytophthora ramorum. And now you really need to go the extra mile, and any Phytophthora, in my opinion, should be sent in for diagnostics. I always go back to actually emerald ash borer, and what would have happened if somebody would have identified that first tree showing symptoms. How much different would our landscape actually look like? And yes, this is going to be expensive, but if you think that it's expensive, I think you really want to consider how truly expensive not knowing is. And here are some photos. These are old photos from the Oregon Department of Agriculture, where they were actually I guess they were burning, burning part of the forest in Cook County, Oregon, and here's the after effect. And they were trying to stop the spread of sudden oak death, and it turns out it was too late. Here was a nursery inspector in British Columbia, Canada, where they were actually packaging things up and removing them for disposal, and it cost well over $1 million. So the $50 to $60 for diagnosis is just a small issue. Why is it so expensive, and it could be even more expensive? Well, this is what all of these Phytophthora look like. It's pretty nondescript, and so you can't just look at them in a microscope and say, oh, I know which species of Phytophthora it is. You have to go through and extract the DNA and run PCR and sequence it, and then do some genetic analyses to figure out what it is. And usually you have to do this with multiple genes. You don't have to know this. I just want you to know that it's expensive, it's slow, and it's time consuming. But it's essential if you want to make sure that you don't actually have Phytophthora ramorum and instead have one of your more common Phytophtheras. So this comes down to what Phytophtheras do we have in addition to sudden oak death, and a lot of it depends on where you're looking and where you live. So a study actually done by Colleen Warfield and Mike Benson showed that there were only 6 species of Phytophthora that they isolated, with almost 600 isolates coming out of nurseries. So they looked in a very small, not a small area, but in one type of area, and came up with 6 different species. This is very different than what was found in Minnesota, where they actually identified fewer isolates of Phytophthora, but came up with 11 Phytophtheras, some of which were never identified because they just, it was simply unknown. And then we did this work in Indiana, and what we ended up finding with, again, even fewer isolates, we found 13 species of Phytophthora, and lo and behold, some of them were actually hybridizing with each other. So this is sort of like how you end up getting a mule between like a horse and a donkey, or I think that I said that right. I'm not an animal person, so bear with me. Phytophthora can do the same thing and create hybrids. So these are some of the reasons we're so concerned, because we don't know what Phytophthora ramorum can do, in addition to the fact what it can already do by itself, what could happen if it hybridizes. So that was an overview on what Phytophthora can do. Really, what do we want to do to prevent it? And this was a picture I took out in Oregon, and everyone should have some sort of plant protection program in place. And so I view trying to protect your plants against Phytophthora sort of like trying to manage the zombie invasion. And so the steps for managing a zombie invasion has to deal with the issues of avoidance, termination, and disposal. And we sort of do this a little bit differently, and I'm hoping what we look at is quarantine, detection, and eradication. But you'll see as you read through there's a lot of similarities. So the first thing we want to do is when any plants come in, we want to check them out and make sure that, they're not quote unquote zombies, that they aren't infected already with Phytophthora ramorum. To do that, we need to go through and detect it, and then when we find it, we'll eradicate it. So here is one of the ways you can go about doing this, and this is in the off season, which is why you can see a lot of other things stored in there, but this is a large production nursery that actually has a quarantine house, and all plants go through here and are evaluated before they're put into, for lack of a better word, the general population. I would especially do this with your, your filthy 5, your rhododendron, your pyrus, your calmia, azalea, and gosh, I already forgot the 5th one. But I would definitely do this with things that are more Phytophthora susceptible. And anything that is symptomatic or test positive, you want to reject, and you just want to keep an eye on things. This is also something obviously space is an issue, so you might want to limit it to high-risk host plants like the filthy 5, or you might want to also just focus on highly valuable plants, but you want to keep those highly susceptible plants together. You don't want to introgress them throughout your nursery and possibly spread not just Phytophthora ramorum, but other pathogens as well. When you're out there and doing your detecting, scouting is another word for it, walking around, you aren't going to find any problems if you're not looking for them. So, make sure you know what Phytophthora you have, and then look through your plants, but don't just focus on your plants. You want to look around at the surrounding areas. So here you can see there's drainage issues going on. If there's standing water, if there's leaky irrigation lines. Other issues to be concerned are, maybe areas that there's a lot of debris accumulation or simple garbage. If you're seeing weeds or algae or slugs, I mean, as we all know, algae is a hazard to people just walking, as are slugs, those big banana slugs. But when you're seeing the associated insects like shore flies, fungus gnats, that should also tip you off that there might be a water management issue. Cultural, this gets to be a little more interesting because we don't have Phytophthora ramorum resistant plants, but we do have plants that are being screened for Phytophthora resistance. And whether it's the Cora series of Vinca that are being produced by Syngenta Seeds, and you can see some of the parents over here doing so much better than everyone else. And this was out in eastern Ohio, and I'm blanking on the name of the research center that was actually evaluating Phytophthora cinnamomi resistant rhododendrons. But there is this work going on, and I think we should support it because it just makes for a better grower end user experience. When you're looking around, one of the big things I see a lot is fertilizer, over-fertilizing. What over-fertilizing does is the excess salts cause the roots to leak. Those leaky roots attract zoospores, not just of Phytophthora, but also Pythium. When you look at something like here where you can see algae and liverworts growing, this media is too wet, and the fact that there is Phytophthora coming on these container conifer seedlings or not container, but this seed tray of conifers is certainly an issue. Whenever possible, creating a barrier between your feet and the soil, a 4 to 6 inch layer of mulch or gravel is important, and making sure your pots aren't in contact with the soil are also exceedingly important. Cleanliness is next to impossible when you're working with plants, in my opinion, but you still have to strive for it. And I can generally go into a nursery, and before I even, look at a single plant, I can tell what I'm going to, or I can anticipate what I'm going to see based on the cleanliness. And, here you have something where there's a cull pile. The cull piles are near the plant, so any pathogens could end up washing into your production area. Pots needed to be clean. You have pallets. I don't want to discuss too much about, treating and disinfecting pots. There is a link we can share with you later about that. But you really, if you start with crap, you'll end up with crap. There's just no other way of saying it. If you do end up getting Phytophthora ramorum, or any Phytophthora for that matter, because I think these suggestions go far, not just with ramorum, but managing any Phytophthora issue, is improving drainage and restricting water movement is so important. Keeping the splashing down so you can't get that foliar stage, making sure you remove those infected plants, because this is reducing your inoculum. And when we say infected plants, we're talking this here, this here, this here, and then all of these adjacent ones to try and reduce the spread. Minimizing access and movement, whether it's on equipment or on employees. These things can easily be tracked by your feet. And last but not least is making sure that you don't take something real susceptible like a pyrus and, putting it into, nearby rhododendron. I would be concerned about putting it, nearby something like lilac here in the Midwest, because lilac actually supports a lot of spore production for Phytophthora ramorum and other Phytophtheras as well. So these are some of the things to keep in mind. And maybe you want to put like introgress them with perennial grasses or something that would not be a host. So here is the much promised sanitation for nursery and the link there, and I'm sure Robin and I can get that to you later. And that just goes through what you need to do to steam your soil, pots, and everything else in case you do have any Phytophthora, so you can remove it. So this is my favorite part of the talk, and this is where we talk about fungicides and how we manage them and how we manage pathogens with them. So it's really important when trying to work with Phytophthora. We have so many different fungicides now that are just for Phytophthora that we actually pay a lot of attention to their formulation. I've worked with growers who think they're going to save a little bit of money by using what is essentially a less expensive formulation, which turns out has less active ingredient, and as a result doesn't provide the control that they actually need. The next thing is how these are actually going to be used by way of, are you using it as a spray or as a drench, or is it a granular that you're incorporating into your media? A lot of how successful something is has to do with how it's actually applied and how you're applying it. And then finally, some of the premixes and when you may want to use those or actually avoid them. I think the most important thing is to really focus on, if you're going to be successful with fungicides, and this is for any disease, not just Phytophthora ramorum, not just Phytophthora in general, but any disease, is you really want to be preventative. And there was one large study with Phytophthora cryptogea, and what it showed is that if you apply it after inoculation or a day or two after infection, it's too late. If you want it to be effective, you want to make sure you have the fungicide in play before the pathogen ever gets in contact with the plant. Now, in this study on Phytophthora cryptogea, the biopesticides used and the phosphite salt, they were not effective, but there have been other studies that have had different results. In this study on Phytophthora cryptogea, Fenstop, Adorn, Mycora, and Segway all gave the best results, and these are some of the newer, more expensive fungicides, and we'll talk a little bit about that here. So a lot of which fungicide you're going to use has to do with where you can use it. And so we have this handy dandy table that we can hand out to you at the end, or send you a link, or send you the PDF, where I tried to show what you could use in rotation, so we have all the frac codes, and then where you can use these things. So something like Fenstop, which worked really well, can only unfortunately be used in a greenhouse. Same with Banrot, which is a granular formulation, but something like, say, Broadform or Mycora or Mural or Orchestra, you can put it in the greenhouse, in an interior scape, a landscape, a nursery, or a shade house.
Janna,
Make sure, yes.
I'm sorry, I have a question here for you.
Awesome.
Are all oaks hosts? Why isn't Quercus alba listed on the USDA official host list?
So the red oaks are believed to be more susceptible than the white oaks. I do know that there were pathogenicity tests performed, and I would guess that Quercus alba was not found to be as susceptible, or they had a hard time getting disease for ramorum. I have seen other Phytophtheras affect white oaks, so it could be a fluke. The big focus, though, for the oaks are actually the red oaks and the live oaks, since that's kind of a weird category. So that's a great question.
Thank you.
Sure. So here we have the list. We will be sharing this with you if you so desire. One of the things about Phytophtheras, which to me as a plant pathologist is fascinating, and for those of you who are growers or arborists will probably find maddening, is how different they respond. And this shouldn't be a surprise, because we wouldn't expect a dog, a Chihuahua, to act the same way as a Husky, as a Great Dane, as a wolf, but all of them are considered in the genus Canis. So I don't know why we would expect all these different species of Phytophthora to behave the same way, but this was an older study that actually showed that Stature, Fenstop, Segway, and Subdue all gave very good control against Phytophthora ramorum. But sometimes with other Phytophtheras, here you can see the Drexleri did not get good control with phosphite. And here you can see that Cryptogea didn't. So these are things to keep in mind, and once again, why you want to know your Phytophthora. Another issue with your Phytophthora and your fungicides is it doesn't just matter which pathogen you're working on. In this case, we're looking at different pathogens and how different fungicides control it, but it also matters on the pathosystem you're working with. And this kind of goes back to, why is Quercus alba not on the list? Well, it probably wasn't, wasn't a very good host in the studies that they used. It could be an accident, it was one experiment, or it may in fact just not be a very good host. Here you can see 3 very good hosts of Phytophthora ramorum, and these are Pyrus, Camellia, and Viburnum. And you can see how these different fungicides all performed, differently depending on the host. Pyrus, I would consider the most susceptible. This would be similar if we were also looking at rhododendron. And you can see that most of the fungicides did not perform as well on Pyrus as they did on Camellia and Viburnum. So using something like Subdue Max to control sudden oak death might not be your best strategy, but using Segway or using Stature or using, Fenstop, which is not labeled outside of the greenhouse, and another fungicide which isn't available, Zoxamid, these provided the most consistent control. Heritage, great fungicide, provided very variable control depending on what your host is. Empress, which is a soil drench, provided really good control against all 3 hosts. So again, this is just some of the variability that we're dealing with. So keeping in mind how susceptible your host is and what you're trying to do. Protecting, though, is always going to give you better results than waiting until after infection has occurred. So recommending a bunch of fungicides and not talking about cost is kind of only giving you half of the conversation. And there's a lot of fungicides out there. One of the newest ones is Segovis, and unfortunately they really need to do a price adjustment because it can be so expensive. If you were to use 38 ounces, that would be over $1,000 per treatment. So you really want to think about what you're going to use, where you're going to use it, and how you're going to use it. Obviously, certain products are going to be more affordable than others. I would say that within all of these different price ranges, that there are effective fungicides, and one of the things I really want to caution against is, for example, something like Segway and Segovis, even as you can see here, you don't need to use as much. So it might be worth your while to use what is by far and away one of the best products for Phytophthora, since you're only using 0.6, less than half an ounce, compared to Segway where it's 1.5 ounces. It also gives you longer control in the case of Segway, which is why I'm such a big fan of this product. You're looking at 14 to 28 days of control. And so when you break down that price, even though the bottle is expensive, per treatment, it could be as little as $14 a treatment, which puts you in line with a lot of the less expensive fungicides that you're using, but it gives you more control and it's more effective. So these are some of the things you should keep in mind when you're actually making your decisions and what you want to do. What am I trying to protect? Am I getting it on in time? Is this host super susceptible? If it is, I probably need to use a bigger gun. If it's not super susceptible, maybe I can dial it back and go with some of the phosphorus acids or Subdue Max or something like that. So then it comes down to formulation, which can get to be a little bit confusing. I became aware of this when I had suggested Subdue and the grower was applying this granular formulation of Subdue, which is just 1% mefenoxam. Plus it's a granule, so you can imagine if you were trying to apply this granular and control a foliar blight, all your granules are going to fall off your leaf and you're not going to get very much control. Granulars are great when you're incorporating them into the mix, but other than that, I don't recommend the use of granules. Using something like Subdue Max or Subdue water-soluble packet, this is a much higher rate. This is for a much greater volume of fungicide that you're spraying for your bigger nurseries. This would be for your smaller nurseries. These you could use as either a spray or a drench, which is really important if you're trying to control the foliar stage of this disease or a root rot stage of another disease. So I think I pretty much said all of this. One of the things to keep in mind with this fungicide and with the phosphorus acid and the alliette is they are systemic fungicides. So these things are xylem mobile to a certain degree. Many of them are not. You need to be careful, and again, check with that label, but Subdue is somewhat xylem mobile, and Agrifos, Aliette, and all of those are very mobile. So getting good coverage and making sure it goes throughout the plant is a lot easier. So one of the things we need to think about is that all of these Phytophthora pose a risk to our industry and our ecosystem. And this here is actually a chinkapin oak, which is actually considered a host species and one of the growers I've had the pleasure of working with. We need to try and make sure we don't get any more established than we already have, and taking precaution. I don't mean being paranoid. What I really am saying is, let's really just be vigilant and maybe overly vigilant on our rhododendron and our pyrus and kalmias and things like that, and just keep the regular level of caution that we've always had. We need to keep these surveys going. We need to make sure all of our Phytophthora are identified, so we don't say, oh yeah, that's just Phytophthora citricola, and it turns out we were wrong, and suddenly we've introduced ramorum all throughout the eastern part of the Mississippi. And I do think we need more studies on the eastern plant species, all of our oaks, and make sure we really understand what is going on. It may be that it isn't particularly virulent, but it could be, and I'm just throwing this out, I don't know the data or the research, it could just be a minor root rot in many instances. So we need to have some good data, scientific data, in which to inform our decisions instead of guesses. So to help you with the diagnosis, and I'm just going to wrap it up with this. This is one of my favorite photos from John Hartman, and this was predating Phytophthora ramorum. And you can see here, these are taxus, and over here we have viburnum. And I don't know what this Phytophthora is. John didn't know what this Phytophthora was. Again, this was a time when we were happy to know it was Phytophthora. And what we found was that you could see that it spreads there. You need tools to help diagnose these problems. So Cliff Sadoff and I have gone and produced the Purdue Tree Doctor, for those of you who don't know, and I'm guessing if you're listening here, you probably do. You can look up your hosts, like in this case, you can look up what all the possible things that can be causing the problems. You could even enter the name, or if you want to show off, you can search by the Latin, show what a fancy pants you are, and then you can go based on what your symptoms are to start to figure out what's actually going on. And so, whoa. For this one, you can see Phytophthora root rot. Here you can see, on white pine. Here you can see it on yew. I like to refer to this as death and taxus. I know, I'm so funny. And just a number of pictures showing you the different symptoms that Phytophthora can cause. We also try and provide information via the Purdue Landscape Report, and we do have something on sudden oak death when we did find it back in May. Well, we found it in April, but we were able to report on it in May. And last but not least, Cliff and I, more Cliff than me, have been working on the Shrub Doctor and hope to have that out shortly. So please we'll try and keep you posted on that. So with that, I just want to acknowledge Adam Leonberger, who did the Phytophthora work with me, and these were his lab mates, and this is his wife. I won't say current wife, this is his wife, and his still friend, Casey, and Kim. And here's Cliff Sadoff. He's being funny there. And these are just people that I'd like to thank, because none of this stuff can be done. It's not done in a vacuum. It's done with a team of people. So with that, I can take any other questions.
Thank you, Janna. This has been really comprehensive. I do have a couple questions for you.
Great.
Tom Macy asks, we're planning to do stream baiting downstream of high-risk sites in Ohio in 2020 around stores that have received infected plants, properties where homeowners reported potentially infected purchase plants. Any other suggestions on follow-up activity, visual survey of symptoms on host plants in the area?
Oh, that's a great question. So I think the stream baiting is hugely important. I do know from work that was done in Georgia, I believe it was, that they kept having successfully baited it out, they were unable to figure out where the pathogen was. So they didn't see things that were symptomatic. So I think the first and most obvious thing is to continue those surveys to homeowners who might have or who were confirmed to have put these plants in the landscape, and really look at anything that is symptomatic of Phytophthora. And hopefully we'll have the Shrub Doctor app out, and I included every photo of Phytophthora on shrubs I could possibly come up with. And I hope that would help you. And I would say, when in doubt, sending things into the clinic. This is one of those things where I don't think we can be too cautious at this point. I wish I could give you more specific examples of what could be done, but I think the stream baiting and then just the visual observation, setting up and doing a transect, and the problem is you're looking for what is a rare occurrence. But if you shoot me an email, I do have a sampling strategy on how to try to improve your detection of rare occurrences. It's a paper, and maybe that would help you, Tom.
All right, thank you. Can Phytophthora be spread by dead plant material like firewood or leaves?
So I don't know so much on the wood part, but for leaves, absolutely. I suspect the wood, at least when the wood is still quote unquote fresh or green, that could certainly serve as a source. Leaves, absolutely. Phytophthora, depending on the species, and certainly I think ramorum has demonstrated that it's a very good saprophyte as well. So yes.
Okay. If P. ramorum is identified in a retail inspection, is there an effort to track down people who may have already purchased infected plants?
Yes, they have. So we always do the trace back to figure out where the infected plants came from, but we also do a trace forward to figure out where they went, and to try and figure out where it is in the landscape. And at least here in Indiana, we did do that, and then try and mitigate when necessary.
Okay, thank you. Please reiterate what to do if we suspect phytop in nursery stock. Do we catalog all testing?
I think you probably, yeah. I would catalog all testing, because there could be issues down the road. One, somebody saying that you are spreading ramorum, and you can say, no, we're spreading something else instead, which probably isn't a great thing. I mean, like I said, I think it's just better to know. And so I would catalog and keep records. If you know where your problem areas are, it helps with your IPM, because you know which hosts are problems, and you know which blocks or areas you need to be careful in managing, or might have issues with irrigation, with leaky irrigation lines, or maybe it's heavy clay or the water table's high in that area. So I'm big into record keeping. My biggest problem is, after I make all these records, finding the appropriate one. So I wish I had a better answer for that part. Did I answer that?
I think so.
Okay.
Yes, she says.
Okay.
Okay. And she says, thank you. Okay. How long will P. ramorum persist in soil in absence of a host?
That's a really good question, and I don't know of any studies that have addressed that. Other Phytophtheras can persist for several years, and I don't know how much we can extrapolate from different species of Phytophthora, but if you recall, going back, I'm going to just escape here and pull up that photo for you to see. Yeah, my gumball photo. Here you can see, those are some pretty thick overwintering structures here. I mean, this is a pretty thick wall. So if we do have the oospore stage, I think it would easily persist for several years. The question is do we have both mating types to produce this, or do we just have the thinner overwintering structure referred to as a chlamydospore? And I don't think we know the answer to that either.
Okay. There's a question. Could you put up the scouting slide again?
The scouting slide again, sure. Let's see. Oh, too far. Here we go. This one?
I'm thinking so. Yes, she said.
Okay.
Thank you. Okay. I have another question now. You said it could be transferred on leaves. So do you think it could be transferred via the movement of acorns as well?
Oh, I don't know. So I would suspect no. So one of the problems or one of the curiosities with this pathogen is on oaks, it is the terminal stage. So it cannot spread from the oaks, it just kills the oaks. And this is one of the weird things about this pathogen is it's really spreading from your pyrus, from your rhododendron, from your lilac, from your viburnum, and that these other hosts are actually, even though they're dying, they're not spreading the fungus. They're just dying from the fungus that is being spread by understory plants. So I would guess acorns would not be, but I don't know. That's just a guess, and I don't know of any data. I will make a note and see what I can come up with.
Well, thank you. I'm not seeing any more questions. I did get a comment that that sudden oak death has been found in Ohio. So I'm just so.
I believe I had that on the slide that it was confirmed.
Yeah.
So, and again, part of it comes down to, oh, nope, it didn't. I apologize. That should have, I lost a star somewhere. But yes, it was, you are correct. It was confirmed.
Okay. Okay. I have a person who said he came in late. And did you have a slide on non-oak hosts?
Non-oak host. Yes, I did. So this would be the big list from the West Coast, but here on the East Coast, what I did, because I mean, we don't grow osmanthus or arctostaphylos too much, or even, I mean, in the South, they grow camellia. So expanding the list that already was there, these are some of the hosts that we know are hosts for Phytophthora and have either been identified positively as hosts for ramorum or are hosts for other Phytophtheras, and the ones that are in bold were previously identified as known hosts of ramorum.
Okay, thank you. And also just remember, folks, if there's issues that you want to see that you heard but you wanted to get more information on, this is recorded and it will be available to view again here in the next couple of days. On the emeraldashboard.info site under the EAB University page. So you'll have that to check out too. But I think it looks like we're good with the questions. And I wanted to thank you, Janna. This is great. There's been a lot of interest in this and we really appreciate you agreeing to come on and share your expertise and knowledge on this. This has been really helpful.
I really wish I didn't have any expertise. I really tried not to develop any, and I was really hoping I would never have to develop any. So I'm sorry I have to be here.
Too late now.
Even though you guys are lovely.
You're dragged in.
So yeah.
All right. Cliff says, nice job. So,
thank you, Cliff.
All right, folks, I'm going to end the meeting. And remember, you will be getting an email from me tomorrow, and I hope you will answer the survey, and it will also give you some information on how to get a hold of Janna if you find you have other questions. So thanks again, Janna, and I hope everyone has a great day.
You too, Robin. Bye-bye.
Thanks.